So far we've found a way to know the levels or figure out the levels of specific messenger RNA within a cell. So if we have a culture of cells and we do something to these cells, right? We add a drug, we add some virus, we add some microbes, we do something to it, we can then come in and use qPCR, RT-qPCR, reverse transcriptase qPCR, to figure out the levels of a specific messenger RNA and how they change because of what we did to the cells. Now, messenger RNA is nice, but messenger RNA is not the end result of gene expression. The end result of gene expression is gene to messenger RNA to protein. So if we know the levels of messenger RNA, we don't necessarily know the levels of the protein. So how do we figure out the levels of a specific protein within a cell culture and how it changes because of what we do to that cell culture? And to do this, there's a technique called Western blotting. But before you can do Western blotting, you will have to separate all cellular proteins with a gel based on their size. So the way you do that is you use polyacrylamide gel electrophoresis. Or PAGE. Now there's two types of PAGE that you can do. One is native PAGE. In a native PAGE, you do not change the native structure of the proteins. And then there is SDS PAGE. In SDS PAGE, you're going to unfold the protein. So this proteins will be in non-native structures. They'll be completely unfolded. Now, what is SDS? SDS is sodium dodecyl sulfate. It is a detergent that is negatively charged at pH 7. So we're not really interested in native PAGE. So we're not going to be doing this. We're mostly interested in this. Okay? Now what does SDS do? SDS will unfold the protein, in essence, linearizing it. So no matter the protein, whatever size it is, the polypeptide chain will be unfolded. Any dimers, tetramers, etc., meaning protein complexes that are held together by non-covalent means through weak interactions, those will be disrupted too. So you will have polypeptide chains that are unfolded and SDS will bind to polypeptide chains, one molecule of SDS for about each 2 amino acids. So you have a uniform charge to mass ratio, remembering that SDS is negatively charged. Now, the polyacrylamide gel is set up as follows and looks like this. If you look at it from the side, what you'll have is 2 clear glass plates. So these are both glass plates. There could be clear plates also, clear plastic plates. And the gel would be in the middle of them. So this is the polyacrylamide gel. Now, if I look at it from the front, here's my glass plate in the front. And my gel will look something like this. The green being the polyacrylamide gel. Now, you might be asking, what are these indentations right here? The indentations on top, the ones being shown, like for example, here, right? These are wells, and these are the wells where you put your sample in. So you put your sample into these wells containing your protein. Using a pipetter. And then what you do is you put this in a device that applies a positive charge on the bottom, below the plate, and a negative charge on top, a voltage. And remembering, since we treated our samples with this SDS detergent, the proteins will be negatively charged. So now your samples will run in this direction and will be separated based on their size. Now, how will they be separated based on their size? If I have a small polypeptide chain, N-terminus, C-terminus, compared to a bigger polypeptide chain, and I have this charge, this negative charge applied to it because of the SDS being bound to it, so it's negatively charged here, more negative charge here. Polyacrylamide gel is a gel, so it's a viscous solution where there is more resistance being applied to the bigger one than it is to the smaller one. So that the bigger one will have a hard time getting through the gel than the smaller one. So the smaller proteins will run faster through the gel, whereas the larger proteins will run slower. So the proteins will all be separated based on size, with the larger proteins being on top and the smaller proteins running faster and getting to the bottom. And if we're talking about cellular proteins, meaning that we have disrupted our cells and all the cytoplasmic proteins are now spilled out, we treat it with SDS and we put it onto the wells of our polyacrylamide gel, and we apply the voltage and we let it run for a few minutes, we'll get a big smear of proteins. Because, just erasing these arrows, as the samples run through and they get separated based on size, there's a lot of proteins. So the proteins will start to separate based on size, but you'll basically get a smear. Because we're talking about total cellular proteins. This does not give us a lot of information, but this is just the beginning of doing a Western blot. It's the first step. Of course, a smear doesn't tell us much, and usually on one of these lanes you will run a protein ladder. And what is a protein ladder? A protein ladder is just a mixture of polypeptides of specific sizes that you buy from a vendor, and it will have polypeptide chains of specific weights in them and nothing in between. So when you run this on an SDS page, here's my SDS page gel, I've ran it, and let's say one of the wells is this ladder, so we'll just say this is my ladder right here. You will have bands instead of a smear of specific weights. And the vendor will tell you which each band, what weight it is. Here are some protein ladders sold by a company. You can see each different ladder will have different markers, weight markers, and these are on kilodaltons. And you can see that, okay, 245, for example, there, and then down here, 25, and then 11 kilodaltons in weight. So it gives you an idea when you're looking at the protein samples that you ran in the other wells about where the weight distribution is. Of course, again, you have smears because you're just looking at cellular protein. But this is going to be important when we do the actual Western blotting, when we're going to be looking for specific proteins to make sure the specific protein we're looking for is of the right size. So how do we go from this to actually having some useful information about our specific protein, not just all proteins? Because it's very easy to figure out total protein concentrations. That there's a kit that you use out there, you add this chemical to it, and you can easily figure out quantitatively the amount of total protein. But before we do that, before we go looking for a specific protein, let's look at the steps necessary to even get to this point. So the first thing you do, you have your cells. And let's just go with a simple experiment where you have two cultures of cells. To one of them, you did something. You added a drug, you infected them, you treated them in some way, and another one, you did nothing. So one is your experiment, and the other one is your negative control. And you want to compare the two. So what you're going to do is you're going to disrupt the membrane, the cell membranes on all the cells in both cultures. So the first step before you can even disrupt the membrane is you remove the media. That is the liquid that is on top of the cells. You remove the media and you kind of clean to make sure all media is removed by washing the monolayer of cells, remembering that the cells are all stuck to the bottom, right? These are the type of cells that we're going to say that bind to the bottom of the plate. Adherent cells, right? So here are my cells, they're attached to the bottom of the plate. Of course, this picture is not to scale because there'll be millions of cells. I remove the media, I wash the monolayer, a liquid called PBS. PBS is phosphate buffered saline. Saline is just water with salt at the right osmolarity so that the cells don't shrivel or pop. And the phosphate buffer just ensures that the solution is always at pH 7. So essentially you're kind of washing with water, but you're washing the monolayer with a water that is at the right osmolarity and it's also buffered. Now typically you do this 3 times. And why are we doing this? Because the media itself has a lot of proteins, and we don't want to deal with the proteins in the media. We want to know the cytoplasmic proteins. So once you've done that, then you, in some way or the other, whether it's mechanically or by just adding detergents such as SVS, you go ahead and disrupt the cell membrane. So if I disrupt the cell membrane, all the proteins that are inside spell out. So now I have all the cellular proteins. And there's a step that you can do that you basically get rid of all the gunk, which is like the phospholipid bilayer and all that other stuff that you don't need because you just want the cellular protein, but that's not important. We've disrupted the cell membrane, and essentially now we have all the cellular proteins. And then you take your cellular proteins and you treat them with SDS and you run an SDS page. And then you have, here's my SDS page, and we're just going to have 3 wells. In one well, we have our ladder. In one well, we have our negative control. And in one well, we have our experimental sample. We apply the voltage, negative on top, and our samples run. And of course, the ladder will give us nice bands at different weights, as we saw before. And these will be smears of proteins at different weights in both cases. Okay? Now you might be wondering, why am I writing the bands in blue? Because when you run your SDS page, after you run it the right amount of time, you're not going to be able to see the protein with your eyes. There is a step in between where you stain with Kamasi blue. Sometimes it's called Kamasi brilliant blue. It's just a small molecule that will bind to proteins and it will stain them blue, and you'll be able to see it with your own eyes. There are other ways of staining, but Kamasi blue is a pretty popular way of doing it. So at this point, we have separated our sample based on size, and we're ready for the next step, which is to actually do the Western blotting. Now, to do the Western blotting, there's a certain step prior to getting to that point. That is simply that you have to transfer the proteins out of the gel onto a membrane. And that is something that we'll cover in the next video. All right, thank you.